Hello, this is Professor Matt Katroulis of Rio Hondo College. This is the video lecture for Chapter 15 for our Chemistry 110 course on the subject of lipids. So first, let's just start off by defining and categorizing lipids. They are one of the major classes of biomolecules, along with carbohydrates and proteins and nucleic acids, and they are unique among those groups in that they are mostly made up of carbon and hydrogen. In other words, they are mostly hydrocarbon in structure. And in some cases, these molecules might have rings, as we'll see with steroids. So, if we look at the kinds of elements and bonds which make up lipids, we would expect that these are mostly non-polar compounds, and therefore they're not likely to be very soluble in water. And this is a feature that many organisms use to their advantage. So the first thing that we're going to do is categorize lipids based on whether or not they are hydrolyzable. And if you'll recall, we talked about hydrolysis reactions back in Chapter 13. In specific, we looked at hydrolyzing esters, where we added water to esters, and that broke up the esters into carboxylic acids and alcohols. And so therefore it should make sense that many of the lipids which are hydrolyzable are themselves esters. So these hydrolyzable lipids will include waxes, triacylglycerols, which are also sometimes known as triglycerides, and phospholipids. Among those lipids which are not hydrolyzable, we have steroids and fat-soluble vitamins. Before we can really get into taking a better look at the hydrolyzable lipids, we need to look at one very important structural element that's common to the first three sets of compounds here, the waxes, the triacylglycerols, and the phospholipids, and those are going to be called fatty acids. A fatty acid is, as the name states, a carboxylic acid, and they contain a long chain of carbon atoms, typically on the order of 12 to 20 carbon atoms, bonded to the carboxylate group. And you'll recall that the carboxylate group is a carbon with two oxygens, and then there's another hydrogen in the case of carboxylic acids there. We can subdivide these even further into saturated fatty acids, and those contain no carbon-carbon double bonds, only carbon-carbon single bonds, as well as unsaturated fatty acids, which have at least one carbon-carbon double bond, and in nature those are always going to be a cis orientation carbon-carbon double bond. We can further subdivide those if we want into monounsaturated fatty acids, which means that they have a single double bond in their structure, or polyunsaturated fatty acids, which have two or more double bonds in their structures. Among the most common of the fatty acids is stearic acid. Now, if we look at this structure, it's kind of daunting to try to look at how many carbons that has. I will tell you right here, this is 18 carbons. So we have one carbon here at the end, and then 16 CH2 groups, and then finally a CH3 or methyl right there at the end. If we look at this entire structure, we expect that the vast majority of this molecule is nonpolar. There is a very polar group here at the end with the carboxylic acid that could, in fact, hydrogen bond with water. But since so much of the structure is nonpolar, we would consider this to be a nonpolar compound in total. While stearic acid was a saturated fatty acid, here I'm showing you two unsaturated fatty acids. So here I have oleic acid. Oleic acid also has 18 carbons, but you will notice it contains a double bond in its structure. This is in the cis orientation because there is a hydrogen going up on either end right there. This is a monounsaturated acid. Linoleic acid has 2 double bonds in its structure, and again, both of these are going to be cis in orientation. So this is a polyunsaturated fatty acid. I'll refer back to this a little bit later, but I'd like you to make a very clear note of the effect of the double bond. The double bond causes a kink in the structure of the molecule. If we compare these molecules here to stearic acid is not always drawn out in a perfect linear structure like this. However, we would say this is one of the more stable conformations of stearic acid, and you're likely to, if you look at the molecule at any point in time, see it tended to be stretched out this way. That's not going to be possible for unsaturated fatty acids because the double bonds, specifically because they are cis in orientation, are going to add these kinks. And those are going to have a significant impact on their physical structures, as we will see. Also, in particular, although it's not really something that I discuss in this video lecture, polyunsaturated fatty acids like this one have hydrogens right here where the arrow is being pointed, which tend to be very easily removed by oxygen, and then oxygen O2 can add to it, and what that will do is it will cause a fatty acid to become rancid. And so I'm sure you've heard the term antioxidant before. These are the kinds of compounds which need to be protected from oxygen, and why we need antioxidants in the first place. One of the ways that we can classify these unsaturated fatty acids is by an omega number. And omega is the last number in the Greek alphabet, and that means we're going to count backwards from normal. Normally, when we're numbering a carboxylic acid, you know that you would give the carboxylic acid number 1. Using the omega system, we're going to instead count from the other end, so that we would get to the carboxylic acid last. So an omega number is just how many carbons from the end of the chain the double bond begins. These are commonly multiples of the number 3. We need these in our diets to a certain extent, and a lot of them come from fatty fish. So let's take a look at 3 of these. So oleic acid, which I showed you just a moment ago, is an omega-9 fatty acid. 9 because if we count from the end of the chain, 1, 2, 3, 4, 5, 6, 7, 8, 9, that would be the 9th carbon is where this double bond begins. However, if we were numbering it the correct way, of course, it would have been with the carboxylic acid as number 1. Hence the need for the omega there. With linoleic acid, we have an omega-6 acid because 1, 2, 3, 4, 5, 6. 6 is the number of carbons away from the end where the double bond begins. And then with the very similarly named linolenic acid, you'll see that we have an omega-3 acid. All of these are unsaturated. Oleic is monounsaturated, and linoleic and linolenic are polyunsaturated because they have multiple carbon-carbon double bonds. Take a look for a moment, please, at the melting points of these compounds. You'll notice that with oleic acid being the highest melting point of all of these, they still all have melting points below room temperature, which means at 25 Celsius we would expect all of these compounds to be liquids. You'll also notice that as we add more and more double bonds, the melting point seems to drop. We'll look at that in a little greater depth in a moment when we start to look at the structures of triacylglycerols. So the first major group of lipids we're going to look at includes waxes. So waxes are simply compounds that we get by taking a fatty acid and combining it with an alcohol which has a very long carbon chain. A typical alcohol that we see in waxes would be cetyl alcohol, which has 16 carbons. Now, you've already learned that carboxylic acids will combine with alcohols and form esters as well as water. So this formation of waxes isn't anything new, really. It's just that we're using carbon chains that are especially long. If we hydrolyze a wax, all that does is give us back the starting fatty acid and the alcohol. Now, like all lipids, these waxes are highly nonpolar, and for that reason they are very useful in biological molecules. For example, if we look at apples, we know that apples contain a waxy layer on their outside, which helps to keep them protected from water. And of course, a lot of plants have waxy leaves, which help to keep water from leaving the leaf so that the plant doesn't dry out. We see that birds' feathers are often coated with waxes. So it's important in essentially regulating the in and outflow of water for various structures. So here's an example of a hydrolysis reaction for a wax. If we look here, we have a long carbon chain on this side. This is from the acid palmitic acid, which I've drawn around here. And then on this part of the ester, we have also a long chain here. So if I take cetyl palmitate and I hydrolyze it by adding water and sulfuric acid, it behaves as any other ester would, and that is that it breaks up into the carboxylic acid and the alcohol that it is derived from. This, of course, would actually be a little bit tricky, and it's not quite as simple as we see here because we know that these compounds are not at all soluble in water. So there's actually a little bit more to this that we're not showing you, but for right now, this focuses on the major components of a wax hydrolysis reaction that you need to see. We're only going to really take a very brief overview of each of these groups. So that's all I'm going to say right now about waxes, and I'm going to move on to the next group, which are the triacylglycerols. We also call these triglyceride, and I would imagine that term might be more familiar. It's essentially a type of a molecule which is formed when we take glycerol, which is a triol, meaning that it's got 3 hydroxyl, 3 OH groups on it. And then those make ester linkages to 3 fatty acids. So each one of those alcohols combines with a fatty acid to make 3 esters. So we call that a triester. There is no requirement that these fatty acids be the same. For some triacylglycerols, all 3 are the same fatty acids, and in others they could all 3 be different. These are biologically important structures in that they are stored in fat tissue, which are adipose cells, and are used as your long-term energy source. So this is a cartoon structure that shows a very simplified version of a triacylglycerol. So we have the glycerol molecule bonded to 3 fatty acids, which again could be the same or different. Let's take a look at a more scientifically complete view. So let's say that we are trying to produce a triglyceride. This is a representation of the molecule glycerol. It's not drawn terribly well, but it's drawn to make it easy to represent how the structure is going to be formed here. So we have an OH. A point would indicate a CH2. Another point, which would be a carbon atom with 1 hydrogen, because it's got 3 bonds coming out of it, and another CH2 down here. So glycerol can react with 3 carboxylic acids. In this case, they are fatty acids. And each one of these can attach, and then we lose water in each case. So this forms a triglyceride, or a triacylglycerol. And 3 water molecules are also produced, but are not shown to the right simply for lack of space. If we were to take a triglyceride and add acid and water, it would cause the reaction to go backwards, to give us back glycerol and to liberate the 3 fatty acid molecules. Up here you'll see we have a space-filling molecule showing these 3. And it's important to notice how these are lined up. You'll notice that they are kind of nicely lined up, and that is to maximize the attractive forces between each of these chains. Now I know so far when we've talked about intermolecular forces, we mean forces between different molecules. But when we get molecules that are this large, it is possible for the same kinds of forces to actually exist intramolecularly. And so we essentially have like some kind of London forces here in between these hydrocarbon chains within the molecule itself that is helping to keep it this kind of stretched out and reasonably rigid. So we're going to divide triacylglycerols into 2 broad categories. First, we're going to use the term fats. When we say that we have a fat, this means we have a triacylglycerol which is primarily composed of saturated fatty acids. So as we just saw a moment ago, the long carbon chains tend to attract each other rather strongly between the different triacylglycerols and actually internally within the triacylglycerol. And so what that means is that these molecules will tend to have relatively high melting points. And so fats are solids at room temperature. Oils, on the other hand, usually have 1 or more unsaturated fatty acids in them. And because of those unsaturated fatty acids, you'll recall, those put kinks into the structures. And because of the kinks, that limits the extent of the intramolecular attractions that you have within the triacylglycerol, and it also makes it more difficult for triacylglycerols to line up from one to the next. And so ultimately it tends to make intermolecular forces a little bit weaker than what we would expect for fats. And so we expect oils to have considerably lower melting points, and they are going to be liquids at room temperature. So let's go ahead and take a look at a figure from a different textbook. You'll notice here that we have a slice of beef, and our arrow is indicating fat. So if we were to be able to look at this fat on a very small scale, we could see that we have 3 triacylglycerols here. For some reason the oxygens appear to have been cut off on these structures. But you'll notice that they are lining up very nicely here in a very organized way. What kind of attractions are these? Well, here it says induced dipole attractions. Remember, we called those London forces. So there's nice London forces that exist between these molecules. On the other hand, if we were to look at an oil, you'll notice that these double bonds essentially cause the shapes of these molecules to kink, as I said before. They are not able to line up quite as nicely. There are still certainly some attractions between these chains, but not nearly to the extent as within fats. So it's really these double bonds which help to distinguish between the liquids and the solid types of lipids. If we were to try to hydrolyze a triacylglycerol, it, as I said a moment ago, produces the glycerol molecule and the 3 distinct fatty acids. This can be done in acid or base, and here I'm showing you the acidic form of this. So I'm showing you here a triacylglycerol which has three different fatty acids. The top and the bottom one have a different number of carbon atoms, even though they are both saturated, and the one in the middle here is an omega-3 fatty acid. So adding water causes a split right here, and right here, and right here, and gives us back our fatty acids, as well as giving you glycerol, which is drawn a little bit better here than you saw earlier. Now, once these have been hydrolyzed, they can be broken down further through metabolic processes within our cells, and eventually all these carbons can be degraded into CO2 and water, using oxygen also that we take in to produce energy. This reaction can also be carried out in base, although we generally don't see that within the organism. If we were to take a triacylglycerol, such as one we might get from vegetable oil or animal fat, and add potassium hydroxide, we make the potassium salt of carboxylic acids here. And you'll recall, if we go back to chapter 13, that we discussed how this process can be used for making soaps. The next broad category here of hydrolyzable lipids are the phospholipids. So phospholipids are essentially the same as triacylglycerols, with one very important exception, and that is that instead of having three fatty acids, they have two, where we replace one of the fatty acids, one of the external ones here, with a phosphate, which is simply a PO4, not 3- in this case, as we'll see, and bonding that to an amino alcohol. We would technically call this actually a phosphate diester because the way it's joined to glycerol. So an amino alcohol is just a molecule that has both an alcohol functional group and an amine functional group, although in the case of our amines here, they're typically going to be positively charged, meaning that the nitrogen is making 4 bonds. Now let's take a closer look at the structure of a phospholipid. And the only real distinctions we're going to make here is based on the identity of the amino alcohol. So the first amino alcohol that we'll look at is the protonated form of ethanolamine. Protonated simply meaning that a H+ has been added to it. So this is ethanol with an amine group that has been protonated, meaning we've simply added an H+ to this. And if we are looking at a phospholipid that contains this protonated ethanolamine, we call that a cephalin. On the other hand, we sometimes see amino alcohols that have a choline attached. You'll notice the main distinction here between the ethanolamine and the choline is that instead of having 3 hydrogens, choline has 3 methyl groups, and that will give us a phospholipid which is called a lecithin. And so you'll notice that in pretty much every other way, these structures are identical. They both contain a glycerol group with 2 fatty acids, a phosphate. Incidentally, you will notice that phosphorus here is exceeding the octet rule, something that I've essentially forbidden up until this point. But phosphorus, being in the 3rd group of the periodic table, is generally able to do that. Also, you'll notice that it's not 3- but only 1- because the oxygens here are making bonds. So ultimately here, this amino alcohol, which has lost its hydrogen in joining to the phosphate, is ethanolamine, protonated, so it's a cephalin. And then if we look here, the only difference between these is the methyl groups here. So 3 methyls versus 3 hydrogens, and so that's our lecithin. Now, this is a convenient way for drawing these molecules when we're first looking at them, but when we actually look at their structures in biological molecules, you're going to see that they are somewhat different. What we often see is that the phosphate and amino alcohol group is rotated onto the other side of the glycerol. So you'll notice again that phosphate and amino alcohol were bonded in the same way. I've just chosen to draw them here off to the left instead of alongside the fatty acid. And this is because these groups here are charged. The phosphate, you'll recall, has an O- atom on it, and the amino alcohol either has a nitrogen with 3 hydrogens and a plus charge, or a nitrogen with 3 methyl groups and a plus charge here. And we also have these oxygens over here from the glycerol. So these groups are more polar. The fatty acids, as we've seen, are very nonpolar. And so what we're going to do is we're going to kind of reimagine this whole molecule in 2 parts. So this part right here with the glycerol, the amino alcohol, and the phosphate, we're just going to simplify and call that a polar head. The 2 fatty acid chains, which are very long, will be called our nonpolar tails. So this molecule is kind of a dichotomy. It has both a polar part and nonpolar parts. This is going to be very important for their function in making up the membranes of cells, as well as other membranes that are found within the cells themselves. What happens is that we see that the phospholipids will line up next to each other with many, many, many molecules being together to form a layer, and then that layer will be on top of another layer. So we get what is called a bilayer. Now, because polar groups want to be with other polar groups and nonpolar want to be with other nonpolar, it only makes sense that the nonpolar tails of one layer will want to be near the nonpolar tails of the other. And that's essentially what we're seeing here in this picture of a membrane. So here we have, we'll just say that this is the top. Actually, it would make sense to say that this is the top. Those carbohydrate chains indicate that this is the outside of the membrane. And then this would be the internal part of the cell membrane down here. Now, in most areas of the body, the external part of a membrane is largely going to be aqueous, a lot of water. And the same is true of the internal portion of a cell. So we don't really want water to be able to just flow easily, carrying all kinds of ions and substances with it. We need to be able to have some kind of way of regulating what goes in and what goes out of a cell. And this bilayer structure is perfect for doing that. It helps to keep everything compartmentalized. Now, I'm not going to go much more into details on the lipid bilayers here, other than to say that there are also other structures within them. So for example, we have a protein here, and this protein is able to go from one side of the bilayer to another. We'll talk more about proteins in the next chapter. Here is another kind of a protein that has what we call a channel that can regulate what comes in and what goes out of the cell. Here's another protein that is stuck only on one particular side, in this case the interior of the cell, and we could have other ones that might be in other locations. And very important for later, you'll notice that we have here this cholesterol molecule that's kind of hidden in there. I'll talk a little bit more about cholesterols later on, but if you look very closely at the picture, you'll see that every several phospholipids or so, we have a cholesterol unit embedded within. So now, moving to the non-hydrolyzable lipids, we have steroids. In general, the term steroid refers to any kind of a molecule which has this basic skeleton here. So we have a 6-membered ring, which is fused to another 6-membered ring. By fused, what I mean is that it's sharing those 2 carbon atoms right there. And that is also fused to another 6-membered carbon ring, which is fused to a 5-membered carbon ring. As shown, these are all cyclohexanes, except this is a cyclopentane ring. although it is possible in some steroids for these to be aromatic. It simply depends from steroid to steroid. But whenever you see this kind of a pattern here, you should recognize that as a steroid. Fatty acids, I think earlier on I might have implied that they can be somewhat rigid, but not nearly to the extent that what we see with steroids. The atoms which make up a steroid are largely locked into place, whereas the tails of a fatty acid are able to flow somewhat, particularly with the unsaturated fatty acids. And so, as you saw just a moment ago, we find steroids like cholesterol in the membranes of cells because that rigidity acts as kind of like a scaffolding on which the different phospholipids can rely, so to speak. So the different tails of the phospholipids are going to be attracted to the steroid and line up next to them. So in essence, these steroids, specifically cholesterol, help to prevent the phospholipids from just simply floating away. There are many different roles for steroids. Probably the most important is going to be to act as hormones, which essentially send different signals throughout the body. So testosterone, which I'm sure you've heard of, and estradiol are both hormones and both steroids. By far the most abundant steroid in the body is cholesterol, and cholesterol can be transformed within cells into other steroids. So here are 3 examples of steroids here. So here is cholesterol, testosterone, estradiol. Let's make some comparisons here. First, you'll notice with cholesterol that there is this medium-length hydrocarbon chain off the top of cholesterol. Other than that, it's not very rich in functional groups. We have an alkene, and we have an alcohol, hence the name cholesterol. Now, as we know, alcohols can hydrogen bond, but is that going to mean that this compound is going to be very water-soluble? Absolutely not. There is, in fact, one alcohol unit there, but there are so many hydrocarbons here that this molecule would be barely soluble in water. Testosterone, we have an alcohol there at the top, and while we do in fact have another oxygen atom here, I would not expect this compound to be very soluble in water. Estradiol would probably be the most soluble of these 3, and that would hardly be soluble at all. You'll notice, as I mentioned before, that it is possible for those rings to be benzene rings, but it is not a requirement, as we can see with cholesterol and testosterone. I'm sure we've all heard of anabolic steroids, which are injected to enhance muscle growth by some athletes. And the role of those largely is to mimic testosterone, and that signals the body that it should be essentially building more muscle tissue. As I'm sure we all know, that these are illegal, and in sports they are certainly forbidden. Ultimately, what they do here is, since they are mimicking a naturally occurring steroid, you're essentially like pushing down on an elevator button in the body and jamming it, saying, make more muscle, make more muscle. And ultimately, you're diverting resources, you're diverting molecules from other locations within the body, and that can cause quite a bit of damage in the long term. The last group of lipid that I'm going to talk about here, although it's not the actual last group out there, there are other groups that we're just not going to have time to talk about, but these will be the fat-soluble vitamins. And we're going to look at a small list of vitamins here. When you talk about vitamins, they are basically chemicals which you need that are required in small amounts. They are organic. as opposed to minerals, things like zinc or iron, which you need. Some of them are going to be water soluble, like for example vitamin C, also called aspartic acid, which is very polar and easily soluble in water. But some of the vitamins are in fact nonpolar, and therefore they're stored instead in fat tissue, in adipose cells. Now, each of these vitamins, owing to their structure, essentially has a different role within the body, and they generally come from different food sources. So I'm just going to take a quick look at each of these, and I'm not going to go too much into detail with them. So let's take a look first at vitamin A. Vitamin A is essential for vision. It's very similar to the different molecules which occur within the eye to carry the signals from light to the nerve and then ultimately to the brain. So you'll notice here with vitamin A that we have a lot of carbon atoms, a lot of hydrogens, and only a single alcohol over there at the end. So we would expect this molecule to be very much a non-polar molecule. Vitamin D is involved in the regulation of calcium and phosphorus. It is actually not a true vitamin in that we don't necessarily have to consume this in the diet. The body is able to make vitamin D using cholesterol. I don't know if that's clear here, that this is degraded from cholesterol, but you'll notice this long carbon chain here should look somewhat similar to what we saw with cholesterol. So should this OH here. The thing that's kind of missing is the six-membered ring, which was right here, and with another carbon up there, before the body could have broken that down into vitamin D. And so this is important in regulation of calcium and phosphorus. As you also may know, this process is helped out with light. So light is essential in the synthesis of vitamin D. Vitamin E is an antioxidant. You'll recall I talked about how oxygen, O2, can actually attack unsaturated fatty acids, specifically polyunsaturated fatty acids, right in between the double bonds. Those hydrogens can be removed, and then when an O2 molecule adds to it, it can cause the fatty acid to start to spoil. And this is what causes milk, for example, to go sour or rancid. Vitamin E, being an antioxidant, is much more reactive towards oxygen than those unsaturated fatty acids. And so what this vitamin does is it acts as an oxygen scavenger. So instead of oxygen just floating around where it doesn't belong and doing damage, this oxygen up here can ultimately be involved in grabbing it. And then the last one of these we're going to look at is vitamin K. And vitamin K plays an essential role in synthesizing proteins which we need for blood clotting. You'll notice again that we have a very non-polar structure. Yes, there are two oxygens here, but that's not nearly enough to make this a non-polar molecule. Now, as I mentioned, there certainly are other lipids out there which are interesting to study, prostaglandins being yet another type of lipid which is important. However, we're just going to limit ourselves to taking a look at these 5 categories for right now, just so that you have an elementary understanding of these. The more important lipids that you'll need to learn about and their roles in metabolisms will be covered in other courses, hopefully, that you take. So this is the end of the lipids video. Thank you for watching.